Hi all, welcome to the biology class. We randomly pick important topics from various subjects and present the same. This one is going to be a zoology video, and the topic that comes next is multiple allelism from the chapter Principles of Inheritance and Variation. As mentioned in the previous video, any topic comes along with memory tips and applications. In order to remember this concept, we will be first explaining the basic terms, raising questions like what's next, why and where, including simple comparisons and real-time stories. We are starting with the basic unit of genetics, gene. Gene is a unit of hereditary information. Each cell has a nucleus which has chromosomes, which in turn have DNA strands. Genes are the short sections of DNA. Now, one may ask whether a gene is made of DNA or DNA is made of genes. When several genes are put together, we receive a DNA strand. DNA is nothing but a complex molecule that codes genetic information. DNA contains nucleotides, thereby enabling the genes also to contain nucleotides. Nucleotides, along with mRNA, code for proteins. However, not all genes code for proteins. Each gene sits at a specific position on the chromosome called the locus and exists in two copies. When these copies differ, they are called alleles. An allele is a variant of a gene. Genes pass down characteristic traits, health conditions, and even diseases, whereas alleles attribute to phenotypes which are nothing but the observable traits or external appearances and can contribute to hair and eye colors, height, etc. At any specific locus, there can be one or more genes leading to the overlapping of genes, but within a gene, there are basically two different alleles. Whatever we have spoken so far is only about the homologous chromosomes. Homologous chromosomes are nothing but those which have the chromosomal pairs of the same length, same centromere positions, same gene placement. They have different alleles on the same gene at the specific locus. Humans have 46 chromosomes. We will now talk about some interesting stuffs before moving further with the same topic. E. coli has one chromosome, whereas a specific kind of a protozoan has 1600 chromosomes. Humans have 46 chromosomes, 23 from the mother and another 23 from the father. And they eventually pair up. Any homologous pair consists of one maternal and one paternal chromosome. Therefore, in females, there are 23 homologous chromosomes because of the existence of only X chromosome. Whereas in males, there are only 22 homologous chromosomes with the last pair being XY, creating the only one heterologous chromosomal pair. In humans, there are even 48 chromosomes leading to triple XY syndrome. India has got its first DNA bank at Lucknow, and it even has gene banks at several locations. We are going to decipher the definition of multiple allelism from the book. All the genetic segregations abide by Mendel's inheritance, which dictates how traits are passed from the parents to the offsprings based on three laws. Laws in very simple terms will be covered here. As mentioned previously, two alleles on a gene are different. And why is this different? All thanks to mutation. As per the first law, law of dominance, an allele which is more expressed than the other one becomes dominant, denoted in caps, and is called normal or wild allele. And the other one whose expression is masked is recessive. Example, dwarf, tall for dominant allele. Internal composition establishes dominance and recessiveness. As per the second law, law of segregation, these alleles do not mix as one copy is from each parent. As already mentioned, there are two alleles, but when there are three or more alleles on a gene at the same locus of homologous chromosomes, they are multiple alleles, and the inheritance is multiple allelism. Does only one allele exist in any organism? Yes, among bacteria, and there are no major genetic variations among their group. Three allele system forms nothing but the ABO blood groups. What about four or more alleles? Yes, hundreds or thousands of alleles are present on the same locus in case of cystic fibrosis and beta thalassemia. Wait. We missed the third law of independent assortment. During gamete formation, two genes segregate independently of each other, and hence there are separate genes for separate traits. This is just the pictorial representation of whatever we have seen so far. A child inherits everything from his or her parents, be it chromosomes, genes, DNAs, and alleles. As mentioned earlier, the homologous chromosomes have the same lengths of short and long arms, and also the same centromere positions. On the genes, we have the multiple alleles. The specific address of a gene is called the locus. Putting forth more questions and finding answers help in understanding a topic better. What makes an allele dominant and recessive? It is dependent upon many factors, mostly based on the strength of protein or enzyme it codes for. A recessive allele may also code for an enzyme, but it may not be functional. Sometimes a copy may be broken or repaired, making it recessive. Example, MC1R protein is to get rid of red hair. CD2AP is for filtering bad stuffs from the kidney. Genotype and phenotype. Genotype is the sequence of genes in the DNA contributing to various traits. Phenotype is the expression of visible characteristics. Multiple alleles on non-homologous chromosomes. No, it is not possible. Multiple allelism can occur only on homologous chromosomes because in this case the alleles are from different genes, here from X and Y chromosomes. Only at the pseudo-autosomal region the pairing occurs. Yes, there can be more than three, but in conditions of diseases. Example, breast cancer, phenylketonuria, muscular dystrophy, cystic fibrosis, etc. Yeah, a recessive character can also be expressed when there are two copies of the same recessive allele. No, the skin color is not related to multiple allelism. Each gene for skin color has only two alleles. There is a simple, delicious comparison between a food buffet and multiple allelism. Here, the cakes are being compared with L gene, which forms the basis of human blood group system. Like we have different varieties of cakes such as red velvet, white forest and black forest. There are different variants or alleles of a gene, IA, IB, I. The end result is based on the high quantity of specific essence and food colors. Here it is completely dependent upon the dominant allele. Essence and food colors are added at the right stage. Alleles on the same gene occur at the same locus. Real-time stories around us. The gene controlling ABO blood type is labeled as I. Isoagglutinogen, an antigen, or L, notated after the discoverer Landsteiner and is located on chromosome 9. It has three allelic forms IA, IB, I. IA, IB are dominant over I. IA and IB are codominant. IA, A or IA, I is A blood group. IB, IB or IB, I is B blood group. IA, IB is AB blood group. II is O blood group. Eye color in Drosophila. It has got a dozen alleles leading to colors like red, white, cherry, blood, eosin, apricot, ivory, cream. But red has always been dominant over the others. Coat color in rabbits. C gene has 4 alleles C, C, CH, CH, C. Capital CC, black or brown fur. C, CH, C, CH is chinchilla or grey coat. CH, CH is Himalayan pattern. Small cc is white coat. Black is always dominant over the other alleles. Flower color in snapdragons. There are 4 alleles white, red, pink and intermediate. W allele is dominant over the other alleles. There is codominance between R and P and the flowers get spots of both red and pink. Bean population may also exhibit a broad diversity of seed coat hues and patterns.